Good afternoon, one and all. Myself, Pankaj Yadav. I am a PhD scholar at IIT Gandhinagar, working with Professor Dheeraj Bhatia. So, today I will be presenting on dopamine, functionalized red carbon quantum dots for bioimaging, cancer therapeutics, and neuronal differentiation. So, quantum dots are nanomaterials which are basically less in a size of 1 to 10 nanometers, and these are being functionalized with the dopamine. So, before going further, let me introduce this molecule, which has so it's being produced in brain at two locations. So one is substantia nigra, where neurons interact and produce this, and which has a role in movement regulation. As you can see, with lack of that will basically give us a disease which is known as Parkinson's disease. Other location that is produced is at this location, VTA, which is in case of reward that it is being produced. And third, it is helpful in cellular communication. So what happens in communication, the tyrosine here is converted into dopamine and it is released as the synapses, and it is then delivers the chemical message, and this is how the communication is happening. Now, in the normal state that I showed you, that this tyrosine is converted to dopamine and hence being released and recognized by the dopamine receptor, which in case then activates cyclic AMP. Then this is protein kinase H, which has a role in cell transcription and then has a role in signaling. But what happens in case of a disease condition due to environmental stress or mutations? This dopamine is metabolized inside the cells, and hence it is not available for further signaling, and hence the movement regulation stops. So as I was mentioning, this protein kinase A has a gene expression role, and hence for cell survival, and it is also responsible for calcium signaling, which has a role in axon potential generation, and how this is how the signaling happens basically. So recently the potential of these dopamine based materials have been realized for diagnostic and cancer diagnostics and therapeutics. So what the people have used for cancer antigen to diagnose, and it has been, with the previous study, it has been reported that the dopamine-based materials have anti-cancer property, and this is the hypothesis mechanism which they have suggested that when dopamine binds to the receptor, it basically activates an enzyme which in that produces nitrous oxide, which in case activate guanine cyclase, which converts GTP to GMP, and which converts into protein kinase G, and which leads to apoptosis. So this is being hypothesized. Now, with this knowledge, we were synthesizing carbon quantum dots. So we took mango leaves, we dried them, powdered them, and we constituted them with ethanol and took out the extract. And then we refluxed it and rotavapor. So we got the nanoparticles which are red fluorescent, and then we mixed with dopamine and we dialyzed to get free with the free dopamine, and then we lyophilized to get the powdered dopamine functionalized carbon quantum dots. Now this was characterized, the UV peak shows the absorbance spectra, the fluorescence spectra at 670. FTIR peaks shows the functional group on surface, that is alcoholic, OH and carbonyl, and the AFM shows the size of 10 nanometer and height of 2 nanometer. The DLS here shows the solution phase size around 2.5 nanometer. This was in ethanol. This powder XRD shows the amorphous nature of these quantum dots. Now what we found out that this molecule is basically positively. This molecule is positively charged and these are negatively charged. So we then mixed with this, and we see that the negative charge of this being neutralized with the increasing concentration of dopamine, and we saw as the size increased from the QDs alone was 37.8 nanometers. So dopamine alone was 0.728, and we could see the loading of dopamine on the surface, and this is the AFM result, which kind of forms a honeycomb structure as compared to the free QDs here. Now with this, what we used is we used the fluorescence of these two images. So here you can show this is the tissue sections of mouse. So this is the control. Here DAPI labels the nucleus, QDs is the red fluorescent quantum dot, and this is the merged image. So with the increasing concentration, the uptake of these QDs increases, and this was done with this, and what we found is that these were endocytes more in the brain. Similarly, what we saw that in zebrafish, this was an animal model, the QDs alone had no significance as compared to control, but the dopamine increased the uptake of these QDs. Next, what we checked was the cancer therapeutic. So for cancer therapeutics, so this is DAPI labels the nucleus, phalloidin labels the cytoskeleton, red channel is our QDs, and this is the merged image. What we see is when increasing concentration of QDs, the uptake increases, but what happens in the conjugate, that you will see that the cytoskeleton structure of these cells destroyed. These are breast cancer cell lines. So what we see, what this conclude is that these are basically anti-cancerous. Next, what we see, so to prove this, we took non-cancerous cell lines, and what we see that with the increasing concentration, there was an increase in the QDs. And here, the dopamine basically, there was no change in the cytoskeleton structure, but the dopamine enhanced the uptake of QDs in the normal cells. Next, what we did was we checked the neuronal differentiation. As I mentioned the role of neuronal, that Parkinson's disease because of this. So what happens? So we took conjugate, QDs alone, dopamine, and retinoic acid, which is a positive control. After 10 days, what we can see that these the neuronal cells get differentiated, and we can see a fully differentiated neuron. There was killing in case of dopamine, and it is being reported in literature that this dopamine reacts with the cell culture media, and it's produced basically hydrogen peroxide, and that's why the killing. And retinoic acid was used as a positive control. And we image these, and what we see is that we compare the dendrite length. If you can see his here, this image is here. So there was not significant difference in retinoic acid and QDs, which confirms our hypothesis that basically it produces. However, there was no differentiation in case of QDs, and retinoic acid was used as positive control, and dopamine, as I mentioned, it killed the cells. In next, what we saw was whether we differentiated their neurons, and we saw the uptake. So in case of basically dopamine enhanced the uptake of these QDs as compared to the QDs alone. And next, what we checked was the cell migration and wound healing assay, in which what we did was we basically did a scratch assay, and we saw that as compared to the control, there was significant wound closure, approximately around 80%, as there was no significant difference in the conjugate and the QDs alone. So with this, what we conclude is that QDs alone were used to image cells, tissues, and animal zebrafish, and the conjugate were used enhance the uptake of QDs, killed the cancer cell, and promoted neuronal differentiation. And I would like to thank the authors and my guide, Professor Dheeraj Bhatia. Thank you. So the terminology, so we call it 1 to 10 nanometer, 1 to 100 nanometers a nanoparticle, but so we termed 1 to 10 nanometer size as quantum dots. So that's thank you.
Our next presenter is Dr. Krishna Mohan Jinka.